Isabella, your turn.
Thank you, Adrian. Thank you for the very good work on moderating until now. So we start with the second part of the workshop. We are only 5 minutes late on the schedule, so it's fine. Don't worry to accelerate your talks, but please stick to the time allocated. So the first one is an update on ovine FAANG by Brenda Murdoch. Brenda? Thank you.
Can you see the right screen?
Yes, and we can hear you.
Okay, perfect. Well, first of all, I want to thank the organizers for the invitation to talk about the sheep or ovine FAANG project. And I'd also like to thank the organizers for their resilience in getting this workshop actually going, since we weren't able to meet in sunny San Diego. And I also want to thank all of the people that contributed to this project. So as you can see, there's a large list of names, and the work that I'll be talking about is actually the work from all of the people that have contributed to this project. I may or may not be able to advance my slide. Okay, so the Human Genome or ENCODE project has done very well in showing us that not all genetic variation that is associated with biological changes are within coding regions. But as you've heard a lot of talks about a number of different species today, I think it's been obvious that there's actually very little known about regulatory elements in sheep. And in the era of growing population, sheep are actually a globally important species, and particularly in developing nations, this is the species that many people rely on for to sustain their food systems. So identifying gene regulatory elements throughout the genome in sheep is actually pretty important. And maybe more importantly is understanding how these regulatory elements contribute to important traits that people need to select for or against in order to help them have stable food and fiber production. And then lastly, biological understanding of gene regulation is pretty important if we want to start predicting genome to phenome, in particularly in different environments. And so really just trying to get a better understanding of gene regulatory regions in sheep, I think, is pretty important. So unlike other FAANG projects, the tissues that we used for this FAANG project all came from the reference genome. And in order to do that, we actually had to first update the reference genome. And what I'm showing you here in this table is a previous reference genome, OAR version 4, as well as the one of the newer, Rambouillet version 1. And when we started the FAANG project, that's what we did is we actually updated to version Rambouillet version 1. And as you can see, the size of the contig N50 is actually much better than the previous reference genome, as well as the number of the contigs needed for this assembly to capture all of the information in the whole genome is dramatically reduced. But even with this, and in the last 4 or 5 years, the technology has changed a fair bit. And so we thought it was pretty important to even continue to update that. And so I'm happy to inform everybody that there's actually a new reference genome. It was released in 2021, and the paper is just released last month. And so I think you can see, even though Rambouillet version 2 was significantly better, we've made huge strides in version 2.0. And this is pretty important when you talk about a number of assays that rely on mapping data back to a reference genome. To just talk about the FAANG assays that we used in particular for the FAANG project, to look at open chromatin and chromatin accessibility, we used ATAC-seq. And the numbers that you see here in the brackets are the number of tissues that we analyzed for that particular assay from that reference genome animal. So for ATAC-seq, we looked at 33 different tissues. And for ChIP-seq, we looked at 47 different tissues. And much like other people have described earlier, it was a bit of a challenge to get ChIP-seq working for all these different tissues. And here we looked at these four common histone modifications. Also, we had whole genome sulfite sequencing from 8 different tissues and reduced representation by sulfite sequencing for 51 different tissues. And additionally, we had RNA-Seq for 60 tissues and CAGE for 51 tissues. And just a brief overview of the bioinformatics pipeline that we used is we used pretty common quality control, fast QC and trim galore, and then very, again, common pipeline for mapping Bowtie2 and then BWA to align. And for the regulatory elements, we used Max2 and CISR. And then finally, overlaying these different tissues, we actually used Chromatin and then head tools to intersect. So I described on the previous slide, we actually did a number of different assays, but for the purpose of this talk, I'm actually going to just focus on the ATAC-seq and the ChIP-seq. The CAGE data was actually previously published on Rambouillet version 1, and we're in the process of remapping all of that to version 2. Just to give you a bit of a highlight from the ATAC-seq data, as you can see, I'm just showing you here 4 tissues, the jejunum, the lymph node, reticulum, and cerebral cortex. And as you can see, most of the signal centers around the transcription start sites. And if we take a look at all of the different tissues that we performed ATAC-seq on, what you can see, I think, is everything does center around those transcription start sites for the most part. We do see some variation in the peak count frequency of these different tissues, but overall, we're happy with our ATAC-seq results. So to think about the question, where these ATAC-seq signals lie, again, I'm just going to show you 2 representative tissues here, the jejunum and the reticulum. And I think what you can see in the jejunum is that most of that ATAC-seq signal is in promoter regions. So the very cis promoter region in the dark blue, and then a little bit more distal promoter regions in the light blue take up about 50% of the ATAC signal. And if we look at the reticulum, the results are a little bit different. So we see about 25% in those very cis and a little bit more distal promoter regions, and we see a lot more signal in the distal intergenic region, as well as other extra intronic region. So again, we've done this for all of these different tissues, and I think you can look at the profile here, although it's not quite as detailed when we think about comparing all of the different tissues. But for the most part, we see some very similar profiles from those more distant promoter regions. We see a little bit more variation in the very cis promoter region in some of these different tissues. Next, we wanted to look at a pairwise intersection. And so what I'm showing you here is a heat map. The dark blue is the smaller amount of fraction overlap, moving more into the red, which is the greater amount of overlap of these signals. And then the blue bars show the number of peaks, and then for all of the different tissues that were examined. And I think if we just point out a couple different things, what we're seeing here is the gastrointestinal tissues have a lot of overlap here, whether it's, and the signal is really coming from the ileum, duodenum, jejunum, gallbladder, to a slightly lesser extent, the lymph node here, as well as the ileum Peyer's patch. And all of this is overlapping with the spiral colon. So we're seeing good signal, biological tissue overlap, which we might expect. And again, another example of this is muscle tissues here, where we see longissimus dorsi and biceps femoris and diaphragm overlapping with the soleus muscles. So I think our signals are pretty good, and we are seeing what we expect to see or what we might anticipate seeing. Next, just to get a little bit into the ChIP data here, and what I'm showing here is the assay that we did, correlation assay, to look at the different amounts of chromatin states. And we used 9 chromatin states to represent the data. So here you can see all of the different tissues, the 47 different tissues that we did CRISPR on, and point at which they have the least variation is 9. So that's what we use to move forward with. And I won't spend too much time on this slide because you're all very familiar with this, other than to say that we chose not to put the ATAC-seq in these chromatin states. So these chromatin states are really a reflection of the 4 histone modifications that we looked at. When we do look at where they lie across the genome, what we can see is that we actually get just under 1% of active enhancers and flanking active enhancers, and then a little bit more, just under 2% of enhanced and poised enhancers, and then more signal from the repressed and weak repressed enhancers. And then it's a little bit hard to see on here, but the quiescent states actually are slightly different in these 2 middle methylation marks, but most of the genome is we would consider quiescent. Very much what we would expect to see. And then when we overlay this chromatin state with our ATAC-seq data, we see that most of the transcription signal is in open chromatin, and most of the repressed signal is actually not in open chromatin. And so we can start laying and layering these data sets over top of each other to support each other. I wanted to just move on to some of the methylation data, and this is data that was generated by Colton. He just gave that sort of lovely little lightning talk here. And so what we see with our methylation data is we do see a lot of overlap with our GI tissues, as well as a little bit more common in the ruminant stomach. And then finally, a lot of overlap with our methylation in the muscle tissues. So I think we have a pretty good data set here that we can start, again, overlaying on all of the other data sets. And when we look at hypomethylated regions, so those are regions, just to remind everybody, that have low methylation, we see a lot less methylation in those transcription start sites, as well as those active enhancers. And then when we look at hypermethylated regions, what we see is actually those repressed enhancers, poised enhancers, and bivalent flanking have a lot more methylation than the active enhancers. And then just to overlay our RNA data, what we can see if just looking at this graph here, which is showing you transcripts per million on this axis, and then the different states on the bottom axes. Most of the transcripts that we're seeing are in those active enhancers, and we don't see very much transcript overlay with those repressed regions or those bivalent flanking regions. And then again, just overlaying that with the ATAC-seq data, we see a large amount of transcript in the open chromatin state and not very much transcript in non-open chromatin states. And here we just wanted to look a little bit more closer at some tissue comparisons of active enhancers. And I know this is really hard for you to see, so I'm going to blow it up for you. But these are the 47 different tissues here that are lined across here as well as across there. And this is a pairwise comparison. So if you could see, you'd see the first and most similar tissues with active enhancers is the abomasum pylorus with the abomasum. Because this is a little hard for you to see, I'm just going to blow it up. So here again, the pylorus of the abomasum with the general abomasum has a 90% overlap in those active enhancers. And the ileum pairs patch with the ileum also at 90% similarity between those active enhancers. And then when we look at the skeletal muscle and longissimus dorsi muscle, so about 90% overlap between those active enhancers. So I think we have a pretty rich data set here, and we can start looking at tissue comparisons between all of these different assays moving forward. And so just to summarize really quickly, we've defined regulatory regions across these tissues and sheet. And in general, active promoters and enhancer regions reside in open chromatin. They have less methylation and greater transcription, whereas the poised and repressed enhancers are not in open chromatin. They have greater methylation and less transcription. And hopefully all of this information can be used to help improve the annotation on the new reference genome that's now available. And so just to conclude, basic understanding of gene regulation and influences of traits of interest is really important for sheep and the world. Understanding the genetic and biological implications of how this might regulate important traits is also very important, so that it can advance global sheep production, support the meat and milk and wool industry, and the needs to feed the world. And so with that, I just want to thank everybody that's contributed to the collection and the different FAANG assays, as well as the folks at Utah State University that helped us actually collect all of these different issues, and the International Sheep Genomes Consortium, which really most of us that work on this project are a member of the International Sheep Genomes Consortium. And then finally, none of this would have been able without support from the USDA in the form of a grant. And with that, I can take questions.
Thank you very much, Brenda, for a very nice talk. And I think I see 2 questions in the chat. We have 2-3 minutes. So one is from Chris Tuggle. Have you developed plans how you plan to display this annotation of a new genome assembly for browsers?
Have we developed plans on how to display these annotations? So there are some works with some kind of lines, tracks, and make sure that these tracks can overlap. Right now, I'm just trying to finish up. So what I guess one of the things I wanted to share is when we did create the new reference genome assembly, we actually went back and remapped all of this data to that new assembly. So we're a little bit behind with regards to that. But, and right now, I think the new reference assembly is only available on NCBI, but we are hoping to make tracks available for people, and hopefully it can be retrieved in both NCBI and Ensemble.
Okay, sorry. And then there is another question by Dominic, not directly related to your talk. Anyway, currently, no public databases, Ensemble, UCSC, iSheep, contain genetic variants, in bracket SNPs or indels, located on the ovine mitochondrial genome. Do you know if this missing info will be added soon, or where it can be found?
Yeah, that's a great question. And maybe I'm not the best person to answer that. I see Peter's up there. I am hoping that we are going to get, I guess, improvements to the annotation, not only the nuclear genome, but also the mitochondrial genome. I do personally have some SNP information for mitochondria. I'm not sure. So if you want to reach out to me, I'm happy to share that. I'm not sure where else you might be able to find that information at the moment.
Okay, so for the moment, there are no other questions, they may come later. Thank you again, Brenda. And I think we can move to the next talk by Jessica Peterson and Ted Kalfleisch on equine FAANG, off to the races.
Okay, so thank you. I'm going to start this off, and we're going to use our time a little bit differently. I'm going to hand it over to Ted after I give you a brief overview of where we're at. The Off to the Races title, I think, was what we used 2 years ago. At this point, we kind of think we're into the home stretch with the initial assays and data collection we planned, and we're looking towards the future. So Ted, please, the next slide. So the Equine FAANG project started in 2016, and we were fortunate to get funding from the Grayson Jockey Club to get us started. Since then, we've been funded by USDA NIFA And we've collected tissues on 2 thoroughbred mares and 2 thoroughbred stallions. All these have gone through whole genome sequencing and then RNA sequencing for 8 prioritized tissues. I'm not going to list those, but we chose these tissues so they could be compared across species, one of the initial goals of FAANG, and then also lamina, which is important to the horse. All those have all undergone FAANG assays, so the 4 histone modification marks. Nicole Kingsley led 2 publications on those data in the mares that I can reference you to. And then Alexa Barber had a poster number 20 on those analyses in the stallions. We've collected CTCF binding data on all these horses on those 8 tissues. And ATAC-seq data is now in hand and being analyzed. Additionally, Xixiang Peng has a publication from last year where he looked at the use of frozen versus fresh tissue for ATAC-seq. So I'll reference you to that paper if you're interested in how those different methods, the different results of those methods. And then finally, the third aim of our USDA proposal was what Eleonora talked about in her flash talk, and also had a poster looking at really cool questions of centromere function and evolution in the horse. So next, please, Ted. So again, I'm not going to go through all the data, but I wanted to just bring up a challenge that we've encountered with our ChIP-seq data. When we started this about 6 years ago, we collected 50 base pair single end data for our ChIP-seq on those mares. That was the ENCODE standard at the time, and that's what everybody was doing. Things have changed, it's become a lot cheaper, and so we collected 50 base pair paired end sequencing reads on our stallion data. Alexa Barber has done a tremendous job working with these data, and then when he went to start to compare our data between sexes, found that it's not that easy. We can't just compare single end and paired end data as if they're the same thing. There's more peaks being called with the single end data, which I think makes sense because there are smaller peaks due to the smaller reads. However, the paired end data are giving us a higher proportion of peaks that look unique to a tissue and a larger average peak size. So in that figure on the screen, those reads that are outlined in red are paired end data reads. The other bars that are not outlined in red are single end. So Alexa is dealing with a lot of ways now to try to account for this technical difference in the library preparation. And it doesn't look like it's going to be as easy as just pretending those paired end data were single ends. So stay tuned for that, but this is something that I think is going to impact everybody across the community, as I'm sure we've all changed some different methodologies as we move through the FAANG process. So next, Ted, please. So I just want to highlight one more time before I hand it over to Ted, that we're really proud in our community that we've really made this a community effort. So in addition to the 8 prioritized tissues that we've had funding for, over 60 tissues were, we call it, adopted. So individual people took money out of their own pockets to pay for RNA sequencing or ChIP-seq of those tissues. In addition, there's been methylation studies, chromatin run-on sequencing studies, different analyses of RNA-Seq reads, small RNA, and now we're moving to iso-seq reads, all with these tissues from these stallions and these mares for the FAANG project. So that said, I'm going to hand it over to Ted. It's going to be a TED talk from now on. So he's going to talk about a way that we hope that these data can become more easily usable for everyone. So go ahead, Ted.
Thank you very much, Jessica. Okay, so what I'm going to talk to you all about today is the notion of democratizing equine FAANG data. So democratizing the data, how can you use it? So all of us are generating our own data. All of us are looking at our data in our tools. How is it that people in the equine genomics community can avail themselves of our FAANG data in their analyses? So bottom line, I don't care what tools you're using. I don't care how you're using it. I just want to make certain that you can get to it and use it readily. Okay, so is our data fair? So I'll just speak about the horse data. For starters, findable, accessible, fair, that stands for findable, accessible, interoperable, reusable. Findable and accessible, yes, we can do that. Interoperable and reusable by the letter of the law, that's true. The data that we have up in FAANG data portal. That's all FASTQ files. You can download those, you can map those, etc. So yes, they are. We know how to work with FASTQ files. You can reuse them. So by the letter of the law, it's true. We converge. I would argue that we converged rapidly on a couple of standardized formats: the BAM, the VCF, the GTF, the BED. But we don't typically store data this way in our repositories, nor do we typically expose them for efficient use. Okay, so challenge usability, right? Again, all of our FASTQ files are up in the data portal. In order to use this data, you download the data, you download the reference, you put them on a high-performance compute environment, couple that with expertise, voluminous storage, compute resources, time, and now you've done your secondary analysis and are ready to put it into long-term storage. This is a barrier to entry for most people. In fact, I think it's insurmountable for most of our community. Therefore, rendering that interoperable and reusable bit largely meaningless for a large chunk of the genetics and the genomics community. You can, of course, put BAM, BED, big-wig files up into a repository, but the way that most of these are designed, they're requiring complete downloads of these data sets, which requires a lot of landing space and a lot of bandwidth, right? So if you're looking to just put these things on your laptop to use them, that can be prohibitive. Okay, solution, I argue, real-time access to processed data sets. Okay, URLs, we know what those are. We know how to use them. Most online tools that we have access to right now, as well as desktop tools, IGV, the UCSC genome browser, the NCBI genome browser, command line tools like SAM tools, all of these can take URLs as input arguments, right? Without doing anything else to them. You can drag, you can drop, you can use. Okay, so we've put together a little system, equinegenomics.uky.edu. I stink at making videos. I've done it anyway. We'll try. It's kind of clumsy. Okay, so IGV, right? We all know this tool. What do you need to do to be able to use somebody's data? You need to be able to load the genome. Goodness, how does one who doesn't know how to build a genome do that? Okay, so within this tool, you are allowed to load the genome from a URL. Okay, we have put up equine FAANG data and resources. We built a JSON file, which is what you need now. You can drag that, you can drop that into this box. You click OK, and it goes, it retrieves the genome, it retrieves the annotation that I've coupled with it. Some people like NCBI, some people like Ensemble. Hey, we've given you both of them. That tool is available for you. Now you want to use the FAANG data with some data set that you have stored locally. Okay, you can load our data from URLs directly. Okay, we'll clumsy. We will go to the RNA-Seq data. I'm a fan of the adipose loin. So we're going to pull that URL in. We click. And there it goes. Just so what's important here is that this is, so I'm interested in kit and coat color, because who isn't? These are the mega bases of data that I need to answer the research question that I have. Didn't have to map anything, didn't have to download tens to hundreds of gigabytes of data, got what I needed in just seconds, okay? Let's imagine then that you want to layer some epigenetic data on top of that, or better yet, a VCF file on top of this. So this isn't a FAANG VCF file, right? Teruaki Tozaki, he sequenced 100 thoroughbreds. He produced a VCF file. I said, hey, Teruaki, mind if I make this a URL and available to everybody? He said, sure. And here it is coupled with all of our data. Okay, so now what if, scroll around, what if now what you might want to do is bring that into a command line tool. Okay, so you can't see the top so well. I've got SAM tools loaded on my Mac. You can drag the URL for that BAM file right into the view application. You can get the coordinate that you're interested in. You got to remove the commas. That's a bit of a problem. Type that straight into more. And directly on my command line, I am looking at the BAM records, the read data for this region. I think that's pretty powerful. And if you want to develop yourself algorithms that analyze these data, you're going to need that. We've also created an API. Again, nothing new here. Just a URL that you can build that you specify the species, you specify the animal, you specify the tissue. You hit go, it tells you what URLs we have and where you would go to get them. Okay, and then one little bit more. Okay, the UCSC genome browser, really not much different here. Right, so you the equine genome. You enter your gene of interest, you add custom tracks. Again, it accepts URLs directly as they are. So we can create track hubs. I'm sure that will be great, that will be useful. But this presentation of the data allows folks to move these data into any resource that they want. And I think that is incredibly important. Again, I don't want to have to know, I don't want to have to account for how people are going to use the data. I just want to give it to them in a way that they can use it quickly and easily. And we submit. And we go. And we see now that our RNA-Seq data is added more or less as BED tracks. That's not terribly great. How would I rather do that? Well, I'd rather see them as reads. You can go down, you can select the squished view, which is the one that I like. And you can take a look at exactly how the reads line up. Again, I think it's a pretty useful, pretty powerful thing. Okay, so we've got the API. So why, so anybody can use this any way they want to. We've put something together that's for people who are maybe a little more anatomically minded. So this was developed by Jamie Norris from here at UK. Now I can just mouse over these various regions in the genome and see what data we have available for it. And let's imagine that I want to create a document where I just create my own little library of data that interests me for some particular project. I can pull those URLs out. I can put them into this document. I can put them into a Word document. I can put them into an electronic notebook. Right. All of this, again, I would argue is a very powerful platform for how one could use this data. So what else have we got going on? Containerized workflows. And Peter talked earlier about how there was another effort that was going on. Bright young student by the name of Jonah Cullen. He's building workflows to do all of these mappings. We asked him if we could make them available through our system. These are singularity containers that can be downloaded again pretty easily and pretty quickly. You go to the git clone, or you git clone the repository, you CD into the repository. Your initial usage is going to download the singularity container. And then from then on, what you do is you put together a tab separated values file of the FASTQ files that you want to map. This is for RNA-Seq. Multiple tissues from the same individual, sample split across lanes, copy input to the TSV directory, and it will create for you a Slurm submit script that you could then run, generate the BAM files that you need, and then you could transfer the data to us. So the advantages of HTTP access, I think, are formidable. And what's more important, I think that it ticks every box in the findable, accessible, interoperable, and reusable abbreviation. So they do not require full data download. These are common data formats, so they're usable across many platforms, many of which don't even exist yet. And for nearly all applications, you can write code to analyze these data. You can build arbitrary data collections all on your own, independent of anything that we have done. And it's immediately amenable to use in a distributed computing environment, which I think is going to become much more important going forward. Bulk data transfer, this is for these secondary data sets, this is a wheel that we did not want to invent or reinvent rather. I just found out this morning that Kentucky is going to get a license to run Globus. So we will create a Globus endpoint for these data so that you have a nice user interface. You can create your endpoints somewhere on your HPC environments in your local environments. You can download this data, these data for use. Now, the URLs, although you could bring them into the command line and use wget or curl or heck, even pull them into your URL bar in a browser and download the whole things, that's not what they're for. And in fact, that use has been, at least on the web pages themselves, you can't just click on them and download the data. You've got to actually move them someplace to do it. We don't recommend a full download through the URLs, but if bulk download or full download of these files is what you want, we will recommend that you use Globus. I'll end there. Our acknowledgements: UC Davis, Si Chung Peng, Nicole Kingsley, Carrie Finno, Rebecca Ballone at the University of Nebraska-Lincoln, Alexa Barber, Jessica as well, the University of Minnesota, Jonah Cullen, Sean Durward-Ackerst, Molly McHugh, the University of Kentucky, Jamie Norris put together that really cool layered view of the horse forest, Dr. Siobhan Laux, and Dr. Ernie Bailey. Our funding, these are the numbers that are important, and I will second Jessica's acknowledgement of all of the people who have contributed, both additional assays, tissue phenotype collector students. This has obviously been a huge effort, which you can all appreciate, but especially the tissue adopters. I believe this is something that was novel to the equine FAANG effort, where we actively solicited community buy-in, and people made it possible for us to sequence a whole lot more tissues than we would have been able to otherwise. I will end there and will welcome questions.
Thank you very much to both Jessica and Ted. So you got quite a few questions. Because we have about 1-2 minutes maximum, if Peter is available, I mean, he put a long comment that is quite interesting, why you maybe could answer in the chat the other questions. Peter? It's not there?
I'm reading.
Yeah.
Wow, yeah.
Because it was a long comment, but I thought was nice to discuss now. Anyway, maybe we can go through the first 2 questions for you. One is from Michael. How do you handle metadata associated with the features? Is there compression and encoding?
Metadata associated with the features. Metadata associated with the files. That's something I think I can answer cleanly. All of these files have headers with which we can do just about anything we want. Obviously there are format constraints and things of that nature, but we can build a lot of information into the headers that travel with these files that are accessible, that will be accessible always with the file, so they will go with them. Yeah. Okay, so if you agree, because it's 8:30 p.m. here, maybe you can answer by chat the other questions, okay? And I will invite now the next talk. It's an update on the Rainbow Trout genome annotation by Rafet Al-Tobazai and Mohamed Salem. Excuse me for my pronunciation.
So, Rafat? Okay. Thank you.
Thank you. Thank you. Yes. Let me share my screen 1 second. Right. Can you see my screen?
Yes.
Right. Right. First, let me thank the organizer for giving us this opportunity to give an update about our project. Our project is improving a simple and functional annotation of the rainbow trout genome. This is funded by NIFA grant, and the people who work on this is Mohammed Salim, Yaniv Balti, Guango Gao, Hoje Wajo, and Raftal Topasi. They are from University of Maryland, College Park, and USDA National Center of Cool and Cold Water Agriculture, University of California, Davis, and Middle Tennessee State University. I start with first the objective. Objective for this project is we have 3 objectives for this project. The first one is closing the reference genome assembly. We have using 3 steps to do this. The first one, we are going to using PacBio technology by generating 80 to 120 sequencing sequence coverage. Then use the contig to generate use the contig that generate from the PacBio to create a scaffold using a high-C map of the chromatin interaction. Then we're going to anchor the scaffold to chromosome using a linkage map, data collected from, I believe, 5,716 fish and 140 full SIP pedigree family. The second objective is using ATAC-seq to sequence of the transcriptome across vital tissue, such as developmental stage and genetic lines. We have different genetic lines we use for this process. And we used to annotate the reference genome for the coding and non-coding transcriptome, isoform, and alternative splicing. The third objective, we're using RNA-Seq and ATAC-seq and ChIP-seq across various tissue to annotate the genome for chromatin histone and chromatin accessibility. The first objective, see, we have closing the reference genome assembly using long reads, PacBio. Rainbow trout has the first reference genome was published in 2017 by a group from France. And then in 2000, I believe, I think 19, we have the part of our group, which is Yaniv Balti and Guango, they, with other people, they publish another reference genome, because the first reference genome was only 48% was 48% of the base pair was annotated to the chromosome, whereas the second one, which is 81% of, I think the second one's around 89, that reference genome was annotated or anchored to the chromosome, sorry. So to improve the genome reference, we use long PacBio reads, and we have different, as you can see, we have different gene line. We have early line. We have the first one was using Swanson. Compared between those, you can see that we move from 2.17 gig of base pair to 2.3 gig base pair, which is that's quite a bit of improvement, with reducing the contig number from almost 500,000 to only 1,509, and improving the scaffold from 139,000 to only 938. And we have another, which is the group is still working on another one, which is another line, which is Swanson. This is a new one that's improved it from 2.1, as you can see, from 2.1 to 2.3, with the same thing, improving the contig and improving the scaffold at the same time. This is still actually ongoing work, is not finished. And we are actually, the group are working on another 2 lines to try to create a pan genome reference for rainbow trout. The second objective, which is was for gene annotation. For this, actually, we do, as we said, we use ATAC-seq to perform that. We identify 10,640 high confidence transcript that not previously annotated, and we were able to define 1,479 isoform not mapped to the current Swanson reference genome. And at the same time, we find that most of those alternative supplies, either intron retention or exon skipping, where they are accounted almost to 56% of the splice variance. And mostly we use the look for splice variance on growth or muscle aggregation, disease resistance, and stress response and fish migration. And you can see this an example. We have genome annotation for the bacterial cold wall disease. We look at this. If you look at, we have two lines here. We have resistance lines and susceptible line, where on this you can see on the resistant line, where this part here, we have intron retention at the susceptible line. This is actually from one of the protein, which is PTX3. Okay, that's on the red. And where you see that there is quite a bit of the intron pieces is still intron retention on this part. This was not discovered before. Right, and another example for gene annotation, we have for the stress alternative splicing, and we look into different stress part where we look at the high salinity, and we have low temperature, high temperature, crowding, and reused water. And you can see that we have some of the exon are differentially used under all 5 supplies. So you could look at this part where you can see that on the control part, you can see as is less expressed as on the one under stress. So for all type stress, we saw that some of those exon are differentially used more than it's the control part. And the last part of this work, which is the ChIP-seq. Okay, that's the last objective. Excuse me. On this, we start the ChIP-seq. This is still not finished, the ChIP-seq and the ATAC-seq. The ChIP-seq, we start looking for 6 tissue with 2 replicates. And out of those, we have 4 markers. We select 4 markers, the isolation, the KME3, ME1, and K4, ME3. And we have inputs. We have 12 inputs. So totally, we have 48 reaction with 12 inputs. Out of those 48, this again is the preliminary data, is not finished yet. This is just to test the data. And if you hear this morning, you hear that is not easy to do work with rainbow trout on ChIP-seq, and that's what we found. That we actually, some of those data, some of those sequences failed, failed the ChIP-QC, excuse me. And some of those passed. And this is actually is not the amount of peaks or the amount of reads is not enough. So actually we seen one of our problem with rainbow trout is we have partially tetraploid, which is some long piece of data, but part of the genome actually exists on different locations. So is copied on different locations. And that's whenever you do the ChIP-seq, whenever you, one of those steps on the ChIP-seq or ATAC-seq is to filter the read based on the quality. Whenever you map it, we find that's almost 30 to 60%, or sometimes 40 or to 60% is mapped to different location. That will give that. So if you filter based on that equality, you lose almost 50% of the data. So I believe I asked the gentleman who worked today, William, who will present this work, and they said they overcome this by MABIT, which is around 45 million read. That's the only way they have to overcome this problem, the sequence with 40 million read of sequence. So still actually trying to find a different approach to solve this issue. But on general, again, some of those is just the preliminary data, so it's not final data. So this is just the first batch, and you see there's some of them passing QC, some of them failed QC for different reasons. We're gonna actually redo this again. So the same thing is, this is actually the tissues we are using, the brain, intestines, kidney, liver, spleen, and white muscle, and those are all of them vital tissue on rainbow trout. So those are why we select those. And for the second part, the ATAC-seq, and same thing, we have select 6 tissue with 2 replicates for each. Only 3 tissue was past the QC. Even we tried different approach for preparing the library, different approach to preparing the library. Actually repeated 2 different times and tried to get some signal to noise ratio that's be able to continue the process. And only 3 tissue actually passing this out of those parts, which is the spleen, the liver, and brain, where the white muscle and the kidney and intestines failed this, failed the process, preliminary process. So we still actually, this process is still working. So again, this is an ongoing work. So mostly we are still working on this part, okay, that's generating the genome, the second part genome, which is actually as the group on Yaniv and Guango, Pat and Guango, they are working on this as creating a pan genome reference, this on USDA on Center of Cold and Cold Water Disease. And on this part again is done, and we still work on the ChIP-seq. And I've seen that today, the talk, and they recommend use different approach. I think that's when we could try that and for generating the data for ChIP-seq, because this is look like some kind challenge for a lot of people. And I believe for our data is more challenge than other. That's what I've seen. And even with, we've seen that on some tissue has more challenge than other tissue. So like on the kidney was, we could not actually get an actually very good data for the library to go to the second step. So that's one of them says some kind of problem with that tissue. So we are looking for, if you have any of you has any kind of solution or any kind of suggestion, we are welcome that. And why with this,
Rafat, can you please go? Oh, great. Okay. Thank you very much.
This part.
Okay, we're going a bit ahead of time. So there may be a later question in the chat. So have a look at that. And now you can stop sharing the screen. Okay. We have the next talk. It's a new perspective on the immune cell atlas of a chicken by Wesley Warren. Wesley Warren?
Yes, I'm here. I'm trying to get my get going here. Welcome, everybody. So can you see my screen now or?
Not yet.
Not yet?
Yes.
All right.
It's coming. Okay.
Hopefully it's coming now.
Yes.
Technology. All right, we all good? Okay. So this is the title of my talk. I won't delay any further. I know I realized just today that I only had 10 minutes. And so I'm going to try to concisely pull together everything associated with an immune cell atlas that you'll see some details here in a moment. This is the agenda I was going to follow. I'll start out describing what we deem the immune cell atlas for chicken. I'll go over one application using Merrick's disease as an example. I had planned to talk about our Pan-Genome initiative as well, but I will not. And so hopefully we'll hold that for a future talk. So in the past, we've looked at whole genome analysis in my lab and many other labs in this way of looking at the whole genome. And we've looked at chicken in a whole genome sense for many, many years, as shown in this Nature cover in way back in 2004. But we've now shifted to more of a functional annotation, and that's what, of course, FAANG is all about. And so my interests in my lab have also shifted more towards this immune resilience concept shown here for commercial flocks. I'm very interested in trying to understand this lowly heritable trait, and in particular, the host response to a variety of pathogens. And so today you'll see an example of a viral challenge. But in order to look at these types of questions or generate answers for some of these questions or hypotheses, we felt we really needed to have a better understanding of the chicken immune system, and more specifically the cell types that drive that host response to a virus or a bacterial pathogen. So this is the a summary slide of the experimental design that we set up, and this is a collaboration with Hans Chang's group at Michigan State and others. And what you see to the left there in the experimental design is we have what we call a control, and then we have a bacterial mimic, which is LPS, and the B represents broiler lines, and the L represents layer lines. So the goal is to sample 3 birds in each of these categories in this matrix. And then importantly, I want to note that many of the cell atlases have been developed in the past in human and mouse, and others have been based on cell sorting or live cells. This atlas will be solely based on nuclei. And you may be asking, are they equivalent? The literature basically says they are. So for many different cell types, and most of that literature is in human and mouse. So the plan, of course, will be to isolate these tissues. In the lower left corner of that slide, you'll see the various organ systems of the bird that we're going to focus on, and that is the spleen, bursa, circulating lymphocytes, lung because of the viral, the respiratory or viral challenges that these commercial flocks have, and of course, thymus for the T cell component. So the way the process works, we will isolate nuclei with optimized protocols for each tissue type. We will sequence those single nuclear RNA-Seq libraries. We'll go through a series of clusters, and I'll share some of our challenges and some of the results that we've gotten for some of these tissues. We develop these cell cluster-specific gene profiles. That helps us to annotate the cell types. And then finally, once we label those cell types, it opens up a whole downstream types of softwares that we can use to look at the functional or the biological interpretation of these phenotypes that we're interested in. And all the names of some of the people that are the various groups and people that are associated with this project are up there in the top corner under the cell curation team. Obviously, I'm not an avian immunologist, but I'm trying to learn fast, and I'm getting help from these individuals that are much more versed in the literature than I am. Okay. So one of the first things we had to establish, and this is an example for the bursa, is when we generate data on up to 6 samples in this matrix, whether it's broiler or layers, and you have individuality associated with each individual cell, and then you try to, of course, dimensionally reduce that and integrate and control for batch effects, not only between birds, but also between samples and library preps. So we're able to harmonize that data with a tool called Harmony that works quite well. And I hope you're convinced based on this color-coded integration on the far right. You'll see the colors of the samples. Again, this is for LPS, so we have an immunostimulatory and a quiescent state of the immune system. The goal here, of course, is to integrate all and truly understand cell type and not worry so much about the functional interpretation of which cells are responding to LPS. We can successfully cluster those once starting out with a dimensional reduction using the Leiden algorithm. And then you can see here the various numbered clusters. And then we go through the process of using differentially expressed genes that are significantly expressed in one cluster versus all others. And you can see this dot plot matrix here. And what I've zeroed in on is the blue dotted lines is this gene called DENND5B. I had to do a quick look on that. And so let's go through the process of how we might use that marker to try to define what cell type we're working with. Typically, and Chris gave you a really nice overview of how they're doing this in the pig. If you have a mammalian single cell or single nuclei data set, there's a lot of data out there now for human and mouse. And there's a nice database called PangoloDB. And we use this frequently to try to get a handle on what's going on in the bird, even though we know the bird immune system in many ways is very distinct from the mammalian immune system. So again, going back to that example of DENNDB5, or 5B, sorry, if we were to plug this gene symbol into the Pangolo database and infer from mouse and human single cell data sets, what cell type is this? What is the most likely cell type? You can see here, it would tell us it's neurons. And we know, of course, if we're looking at the bursa, we're not looking at a large cluster of neurons, which is circled there, and cluster 2 is what we're looking at here. And then you go deeper and you actually ask, okay, if we look in the mouse and look at bone marrow cells where we know immune cells are going to be being produced, you can see that it denotes that these are B cells based on this marker. So, okay, then the next question is, If we go in and look at that particular data set and we circle here in the left corner the B cells, and then we ask what cells in that whole cluster matrix are producing that gene, and you can see clearly it overlays with the B cells. So by inference, we would go and label cluster 2 in the green there as B cells. Obviously, we're not going to use a single gene marker for denoting the cell type. This gives you kind of a example of the challenges we're working off of for birds, especially with the unique bursa organ system that's not found in mammals. We've done the same process for spleen, and you can see the markers here for spleen. And I want to note that these are preliminary because we're still working through the data. We're cleaning it. We're trying to understand if there's markers that are falsely clustering some of these cell types. So it's a lot of manual curation in this process. So one example I want to tell you about with the lung. So again, we've done the clustering and cell labeling with the lung. Once we have these cell types labeled, we can ask really unique questions about the data. So in this case, we have a whole bunch of virologists here in my group over here at the University of Missouri across the hall that are interested in influenza and other viruses. So we can take our initial identification of the lung cell types in chicken. We can go to databases such as the viral VT Hunter, and it does have some chicken specific data there, but you can see all the data is mostly neuronal or early embryonic development. So this is really not helpful. In the future, we will be dumping our data sets into this kind of database and be really useful for mining cell types that could act as preferential portals for various viruses. So here's an example again where we labeled some of the cell types that express the EGF receptor. And you might ask, well, why the EGF receptor? Well, it turns out, even though it's not considered a primary portal for the virus, it does facilitate some of the virus's success in infecting the epithelial cells in the lung. And if you paint our preliminary analysis of the cell types in the lung, you can see here with the green representing a higher expression of EGF receptor. There are certain cell types there, endothelial cells, fibroblasts, and pulmonary alveolar cells that are expressing this receptor. What's really interesting then is you can take this and start to develop hypotheses. This is one example of paper I pulled and showing that growth factors are important contributors to viral success. So again, this gives you a chance to set up an established hypothesis and work with this further. All right, how much time do I have left? 2 minutes or am I over? She says good, I think.
No, I say in 1 minute. Well,
oh, 1 minute, okay. So quickly, we applied this to Marek's disease, and this is the experimental design. You're familiar, most people are in the poultry industry are familiar with lines 6 and 7. One's resistant, one's susceptible. We did the same except this is live cells. We looked at the differences between those lines in control versus challenge with the virus. And Marek's disease, by the way, is a herpes virus and has oncogenic type properties. And we looked at gene expression. We found really interesting differences by cell type, as well as the scope of expression, meaning higher expression and susceptible versus resistant across all cell types. And then whether it's going up or down is dependent on cell type as well. Obviously, there's way too many genes to start developing, hypotheses all over the place. So we try to use some enrichment techniques. Here's an example where we find enrichment for, the glycolysis energy metabolism pathways. Turns out that some interesting T cell data for LPS challenges, bacterial challenges, not completely relevant to the viral challenge here, but maybe, there may be convergent factors. So the summary is that we can accurately identify cell types. The subtypes are continuing to be refined and revised for Atlas use. And we've only done these 3 cells. As I said, we're going to be looking at thymus and lymphocytes as well. And then finally, we've shown, and we have a paper that's drafted to really kind of define the Marek's disease response in the spleen. And then the last question that a lot of the FAANG members want to know is, how do I get access to this data? We're working with Chris Elsick, who has an NSF grant to really develop tools to mine all the FAANG data. So we're open to strategies and comments from the group here on what you would like to see as part of these tracks. And then I will end there in the acknowledgements. many people to acknowledge, so I won't delay us any further.
Thank you very much, Wes. So you have at least 4 questions in the chat. Because we are running late, so I invite you to reply directly to the chat.
Okay.
And we move straight away to the next talk. Is by Linzao Fang. It's on FAANG pig, a comprehensive catalog of regulatory variants in the pig transcriptome. Okay,
thank you.
You can see the screen. Thank you, Linzao.
Okay, thank you very much. And I think, is that okay? And it's better? Can you see my screen?
Looks good to me.
Okay, great. Thank you very much for the introduction, and thank you very much for the organizers and give this opportunity. And so today I would like to give a presentation on behalf of FAANG GTAC Consortium, but focus on the pig GTAC. So before that, I would like to say the FAANG GTAC is an international collaboration, and I would like to thank all the collaborators and from these institutes and universities. And just a brief background about why we need to do this from the GTAC project. Because in the past, more than past decade, and so we got a lot of genetic associations of the complex traits or phenotypes, both in human genetics or livestock genomics. Using GWAS or population genomics, but there's a big gap between this genotype association with the complex phenotypes, and we definitely need all these intermediate phenotypes to help us to understand. But all these molecular phenotypes are super dynamic and complex, so there are different among tissues, developmental stages, and diseases. So that we need a lot of efforts, especially international effort, to collect this data. And once we got this data, we can develop advanced static models to infer and to understand these regulatory circuitries, and in case to inform us to later to manipulate this, and then we can better we can closer to the precision medicine in humans or precision breeding in animal or plants. So this is a very successful project, and to do this kind of work. So this is the called GTAC project. So here they built the reference of the regulatory elements in about 15 major tissues in humans, and particularly for the gene expression QTLs or splicing QTLs. But inspired by this project, we founded the FAANG GTAC project. So we would like to use all these intermediate phenotypes and to link, actually to open this black box between, the association between the genotypes with complex phenotypes. And right now we finished the cattle GTAC and the paper. So you can get the paper in the bioarchive, and it's recently accepted in a few days. And for chicken GTAC, and you can get more details from the poster by Lu Guang. And today I'm going to focus on pig GTAC. So this project started almost 2 years ago, and hopefully we're going to release all the results in a few months. So all the data has been analyzed in the pig GTAC in the pilot phase, so including over 10,000 RNA-Seq and over 1,600 whole genome sequence to use this data to build the first version of the genotype imputation reference panel. And we collected analysis uniformly for all the published epigenetic data, the high-C data, and the single-cell data, and to annotate all these EQTLs, and also GWAS. So we collected the genotypes and phenotypes for more than 260 complex traits. And this will be used to integrate with the EQTLs. And the last data is the kind of validation data contributed by different collaborators around the world. So first, let's look at the sample clustering based on the gene expression, because this is kind of phenotype and also alternative splicing. So we can see all the samples, if you analyze them uniformly, and you always see those cluster quite well according to tissue types. So this ensures us to look at the tissue specific regulatory variants. And if you look at the panel C, and so we cluster the samples together based on their genetic variants, and we can see the samples in the reference panel, they have the very similar or matched population structure with the RNA-Seq population, because this is the target population. We need to use this as a reference to impute the genotypes that are called from RNA-Seq. And so the huge students and postdocs got involved by analyzing all this data. And another thing, because we analyzed over 10,000 RNA-Seq, this is the largest gene expression atlas. So by using these resources, you can better understand the function of the pig genes. For example, this is just one example. So we can detect the tissue-specific genes, and all these genes can reflect the known biology of the tissues. And by using the gene co-expression network, we can successfully note over 13% of the genes with biological functions. And so then the major objectives of this project is to develop these molecular QTLs as a reference for pigs. So the first step, we need to do the genotype imputation for SNPs that come from RNA-Seq. So at the end, we show that we can do a quite good job for over 3 million SNPs. And we validated this and use a different data size. And all the coordinate rates was quite good. So we think we're probably safe to go further. And then within each tissue, I will remove this. Okay, so within each tissue, we detected duplicated individuals. Actually, the samples are from the same individual by using the RRBS matrix or GIM matrix. So because we got some true data, so we know they are from the same individual. So by using that, we detected to use so we decided to use this 0.9 user cutoff, and to detect if the samples are duplicates, and remove the one with the poor data quality. And after that, we got the enough samples actually for 34 major tissues and with over 40 individuals, and for the EQTL analysis. And then we corrected the population structure and all the unknown confounding factors within each tissue by fitting the peer factors or genotype pieces. The B panel just showing the EQTLs, how they are related to the sample size across the tissues. In general, you can see once you have a larger sample size, you got more power to detect EQTLs. This EQTL for protein coding genes. And the similar pattern can be found for the FAANG mapping. Actually, the independent EQTLs per gene. And so the tissues were ordered based on the sample size from the smallest to the largest. And the red dot is just the mean value of the independent EQTLs per gene. In general, you can see when you have a larger sample size, you got more power to detect the independent EQTLs. And then we conducted the international validation, the internal validation of the cis-EQTLs within the tissues where we have more than 200 samples. So randomly divide them into 2 groups and perform EQTL mapping in each group and validate another group. So in general, you can see this pi values is it's just measure the replication rate. So if they're close to 1, that means we did a good job that we can replicate it in the EQTLs. So in general, we got very good results. And then we conduct the external validation by using the totally independent data. The data we never touched in the discovery population. And when the data size by collaboration with Brittany and from the USDA, where they got 200 whole blood cell samples, where they also have both RNA-Seq and genotype data. So the data were generated at 2 time points. And in general, we can see we got a very good replication rate across all these 5 data sets. And the eQTL file size was positively correlated. And then was also validated for eQTLs detecting other tissues by collaborating with Maria and Daniel. And so they got 100 individuals with both RNA-seq and whole genome sequence data. So in general, we got a very good replication rate. Actually, we can replicate it, the eQTL detected in their data, over 99%, because we, so here in the PICG test, we got over 1,000, more than 10 times the data size than this. But for this tissue, you can see we have less samples than there, and the replication was not that good, but they can replicate the 17%. So that's the replication rate or totally dependent on the sample size you have. And then we'll also use another complementarity analysis, and this is called the allele-specific expression analysis. This will analyze the data at the individual level. So a lot of confounding factors, but not that important. Because it's the individual level analysis. And in general, we can say the effect size of the SNP or the matched SNP, they have a quite good correlation across all the tissues. So I only show here the 2 examples, the muscle and brain. In addition, we also detect the molecular QTLs for another 4 phenotypes, the splicing QTLs and the linker RNAs and the exons and also enhancers. And then let's quickly look at how this eQTL are shared across breeds. So we take muscle as 1 example. We divide the samples into 8 groups according to their ancestry, and like 4 cross breeds, and another 4 is kind of per breeds, Durham, Yorkshire, Landrace. The sample size is different. And so here the pi-value, the replication rate. In general, you can say the Landrace and Yorkshire cross can replicate 90% of the genes that detected in other eQTLs in other 7 breeds. And if you do the cluster based on the 6 eQTL file size, and you can see all the eQTL derived from muscles from different breeds. They'll cluster quite well in the left top, and they were separated from other tissues. And we got the similar results by doing the meta-analysis across all these breeds. You can see the meta-analysis, the effect from the meta-analysis are highly correlated with the effect size derived from the combined population. And let's move to the how this eQTL are shared across tissues. So in total, we got 34 tissues, and we clustered all these tissues based on the 6 eQTL file size. And we can see the biological similar tissues were clustered quite well, like the tissues from the digestive system and the tissues from the brain. And I just want you to see here, because today I think in the previous talk we mentioned that the test were quite different, and here we got the similar thing. So the genetic or regulatory effects are quite different in testes compared to other tissues, and this is just one example, and we found the opposite effect size for these particular genes, how the highly, the same QTLs and in blood and testes, so they have the opposite genetic effect on the gene expression. And let's move to look at the function mechanisms of these molecular QTLs. So we integrated all these eQTLs by different omics data. And the first is just a sequence ontology. So by looking at whether these kind of QTLs are enriched with, for example, the 3 UTR or 5 prior UTR, in general, I can say this eQTL have a high enrichment because it's a log2 transformed enrichment. And the splicing QTL also more highly, have a higher enrichment in the spliced region or spliced acceptor, and also have the highest impact on the sequence. And then we integrated the chromatin states. In general, you can see all these 5 molecular QTLs have a high enrichment in the active promoters. But so here for the enhancers, we saw a very, very strong tissue-specific enrichment. I didn't show the results here. And for the 5 maps, the EQTLs, we can see the first EQTLs, normally the EQTLs with the largest effect size. So they are more likely to enrich with active promoters. But the second, the third, which is the EQTLs with smaller effect, so they are more likely to enrich with active enhancers. And we also checked if these EQTLs are more likely to be enriched in the same TAD that are derived from the high-C data. Once we account for the distance between these the EQTLs with the TSS or target genes, one can say the long-range EQTLs do have a higher enrichment of TAD. So the next also the very important objective to develop this peak GTAs is to understand the genetics of complex traits. So we integrated comprehensively with these molecular QTLs with the GWAS. This is the imputed sequence GWAS. So the huge amount of students got involved in data. And in general, I would like to show that all these 5 EQTLs have the significant enrichment for the GWAS, for the GWAS signals, and the EQTL and splicing QTLs have a larger enrichment. And then we conduct the SMR, and the TWAS and colocalization using a list of the common used methods. And so we can find thousands or hundreds of thousands of the gene tissue traits significant associations. So I just give a very 3 interesting results here. So the first result is the EQTL specific results. Just to focus on the GWAS, this is the average delegate. So the first Manhattan plot is the GWAS. We can see here, this is the peak. And once we look at the EQTLs and splicing QTLs and the exon QTLs for the same genes in colon, we only can say this, the EQTLs, which is the SNPs are regulated, the genes, the entire gene expression were highly colocalized with the GWAS. The GWAS that are derived from our delegate. And let's move to the next in the middle. So this is for the average, the back fat, the thickness. This is the GWAS results. And then we only can say the splicing QTLs for this particular gene in small intestine were perfectly colocalized with the GWAS, not the entire expression of this gene or not any particular axons. And the last example is the exon QTLs. So this is the GWAS, it's the same trace. And we didn't find any signal for EQTLs in muscle for these genes. And no signals for splicing QTLs, but only for signals for particular axon expression that are highly colocalized and significantly. So the next last step, we need to compare with the human GTAs. And in general, you can see the gene expression are quite conserved between the 2 species. They were clustered quite well. So each dot means each sample, they were colored based on tissue types. And this is colored based on the species. And then we also look at the conservation of E genes. In general, we can see in the tissues, the 2 species that share the significant overlap of the E genes in the same tissue, like the EQTLs that detect in blood in pig are significantly overlapped with the EQTL detected in blood in human. And you should know that the sample size are quite different between these 2 species, and so this is a very, very, it is a really good results. and to look at to get this significant overlaps. And then we also found that so we divided in each of the tissue divided the E genes into 4 categories. That's the show E genes in both species, and in another species or only one species. So in general, these E genes show a very distinct sequence conservation. So in summary, the PacGTAS will build, already built actually the largest expression atlas. And this will help us to better understand the gene functions in pig genome, and also develop the pilot phase of the PacGTAS. This is the kind of 5. We developed 5, actually 4. I didn't mention the network, but we did this network QTLs. So we did 6 molecular QTLs, and also we show that these results can really help us to understand the complex trait genetics and also adaptive evolution. I didn't mention results, but we do got a very good promising results for this in pigs. And by comparing reading the human GTAS and pig GTAS at different levels, gene expression, EQTLs, also TWAS level, we do show that pig is a very good medical model for certain complex traits and diseases in humans. And at the end, we are building the open access web portal for the entire community to get access to data. And thank you very much. And at the end, I would like to thank all the collaborators and from all these institutes. Without your help, we can't achieve this. Thank you very much. And that's all I would like to share today.
Thank you very much, Lin Zhao. And we have really to go fast, so you can answer the few questions you got in the chat. The last talk before the roundtable, so don't go away. It's by Ruidong Xiang and collaborators from Australia, using variants with functional significance to improve genome-wide mapping and prediction of complex traits. Okay.
Thanks very much, Elizabeth. Can you hear my talk and see my slides?
Yes. Yes.
Okay, excellent. Oh, hi, everybody. So my talk is about using variants with functional significance to improve genome-wide mapping and prediction. So, so going back to this old slide now, and this old technology about these classic, the genomic selection has revolutionized the breeding industry. Now, I'm pretty sure many of you have seen this graph or something similar many times. Or basically, if you want to do genomic selection, you would have a reference population where you have genotypes and phenotypes, where you derive a statistical predictive equation between the genotypes and phenotypes. And then when you have new candidates, and similar to what humans do in a polygenic score approach, you can get the genotypes, plug them into a predictive model so that you get the estimated breeding value, which is predicted phenotype based on the genotypes. And that allows you to get some sort of ranking of the individuals based on the genetics, and then helps the selection of the top individuals for breeding. Now, this approach is good because it increases the accuracy of breeding decision. You base your selection on the genetic merit, and also shortens the generation tempo because you can make a better decision at much earlier time. Now, at the center of this business is the genotypes used in the training and the validation population. And because the invention of genomic selection or prediction was in the early 20s. So that was the time when animals were usually genotyped on something like a 50K SNP chip. So it is quite reasonable back then to assume that we can use all the markers equally, because there are not many of them can choose, and they're all SNP markers, so there are not many causal variants involved. But now we have millions of sequence variants, and either directly sequenced or imputed. And many studies have shown that if you just go use all the sequence variants to do a genomic prediction, it's not going to be very helpful. So there are several strategies to tackle this problem. One is to pre-select the variants to improve genomic selection. But the question is, how do you select them? Another angle to this issue is that normally we will do an imputation from the SNP chip panel to a full genome sequence, but we do this based on a standard chip, which are basically some random markers selected across the genome. But if we want to do this imputation in a better way, why should we impute from random markers? But why not try imputing directly from some functionally important variants? So a bit more explanation about using the SNP markers in a genomic selection. So normally genomic prediction or selection, they don't use causal variants. So they rather use the markers in high LD with causal variants. It works fine in animals because we have a relatively small population size because the individuals are highly related. But there are issues keeping using this approach because the LD between the markers and causal variants decreases, and especially when you do this prediction across generations and across populations, and that gives us an out of sample, low accuracy of out of sample genomic prediction problem. And so, for example, you train your model in Holstein and predict in Jersey. A similar case when in humans, when you train your models in the Europeans and predict into African ancestry, you get a low prediction accuracy. So we think that if we can use causal variants in this predictive business, then we can improve this because we expect that causal variants would have consistent effects in different populations. So they can provide a more robust way training a model to be based on. The other matter of the using functional genomics in genomic selection is, well, they are there. So they, as many speakers discussed today, and there is functional annotation consortium. There's so many data available in the public, and they will be made public available. This is just a snapshot of what the data set are available in the FAANG, but this is a bit out of date. So I'm pretty sure what we have now is much more comprehensive than this one. And so it will be a shame not to use all this public available resource to help doing breeding. And so, we should use the resources funded by taxpayers and return the benefit back to them. So with these aims and issues and hypotheses, we want to enhance the genomic selection by using the functional information. So we have several motivations. One is we want to enhance the mapping of informative or causal variants by using functional information. And we can also try to use that information to either eliminate useless markers or weigh important markers in a genomic selection. And another point of this business is to try to do this in a de novo approach where we could use the functionally important markers to customize genotyping tools, and then we can start the genotyping in a better place from the very beginning. So in the following talk, I will demonstrate a few cases around this area, the few points that I've discussed here. I want to start these cases with the ChIP-seq peaks. As many speakers discussed today, that the ChIP-seq, the signal and mark promoters and enhancers, and which are important regulatory elements in a genome. So Prowse Wilkins from our group, she studied the ChIP-seq peaks from several cattle tissues. So this is all published work, and for more details, you can go in there. But here I'm just summarizing the 5 marks called from the resources, and they cover about or up to 20% of the genome. So this is not much genome coverage. But then the question is, how are they enriched in QTL? So here is a result summarized from this work. Sorry about a typo, but that should be peak, ChIP-seq peak. So we looked at the GWAS results in cattle, so protein yield, fat yield, and milk yield, and they're all milk production traits, and how are they overlap or enriched with ChIP-seq peaks cohort. And we found they all to some extent enriched, but the highest enrichment is in these yellow bars, which is the mammary gland tissue. And that's kind of expected because these are the milk production traits, and the mammary gland is at the center of, the milk production. So that all makes sense. And now we have many types of functional genomics data set. So we want to evaluate how good are they in terms of helping the mapping of complex traits. So we collected several data sets that we think might be of interest or important to the traits that we study. Now, so for example, we looked at the SNPs under ChIP-seq peaks and all the SNPs related to the coding activities of the genes, the SNPs that are impacting the RNA splicing, just SQTL, or we have a set of finely mapped pleiotropic SNPs, which means that they impact multiple traits in that matter. And we also have SNPs that change their concentration of metabolites in the cattle. And once again, we have a set of SNPs so these are the mutations at the conserved sites across 100 vertebrates. And I think one of the speakers also mentioned the importance of them in the genome. So we think these are possibly functionally important, and we call them class 2. So they are the SNPs with some kind of functional importance. And here we have another class called class 1, which are just from the high density SNP chips. So itself contain about 600,000 markers. So they are they're actually very robust, well used in our breeding industry, and demonstrated they are robust but random markers. So we want to know if we add these functional SNPs to some very robust selected random SNPs, can we gain more power to map causal genes? By the way, so these resources of these functional data sets are presented here. If you're interested, you can have a look at them. And so we want to model this question in a more what we say a refined matter. So we want to use the functional genomics data as biological priors. And what sort of model deals with priors the best? Obviously, it's got to be the Bayesian models. So we fit them as priors in a Bayesian model. So in essence, we can think the genotype-phenotype relationships is our observations, and the biology is our priors. So we can then put them together to adjust our observations, so we get a better posterior estimates. So in practice, we've used a Bayes R model. It's a very established Bayesian model. In essence, it allows a mixture distribution of varying effect sizes. So here we fitted 4 categories of varying effect size. So SNPs with no effects, SNPs with small effects, SNPs with medium effects, and SNPs with large effects. These kind of effect categories are based on the proportion of the genetic variance I explained. Another model we tried is Bayes RC, and now it's just a C on top of the Bayes R model. So it has the same part as Bayes R, which is in the effect groups, 0 effects, small effects, medium effects, and large effects. But on top of that, we have a functional category for each SNP. And so that's what we talked about, the class 1 and 2. So a SNP is classed either they are functionally important or they belong to the high density SNP chips. So here we want to compare if we added the functionally important variants to a high density SNP chip, do they change the mapping precision? And we define that metric as the amount of genome segments needed to explain half of genetic variants. And the second metric obviously is the prediction accuracy, which is the correlation between the estimated breeding value, and so predicted phenotype, and with the real phenotype. So here I'm showing you the results of the mapping precision. Now, so this is a bar plot, and on the y-axis, that's the amount of genome needed to explain half the genetic variants. So basically, you would expect a better result to be shorter bar, because they need less amount of genome to explain half of the heritability. So it's more densely mapped. So on the very left-hand side, we see the high density SNP chip alone, and we use that as a reference to compare. And on the bars on the right-hand side, they're all adding functionally important SNPs from chip-seq peaks, from coding activities. They affect metabolites, they affect splicing, they are conserved, or they are from our fine mapped variant sets affect multiple traits. In fact, our results show that if you add these functionally important SNPs to high density panel, they all have reduced height. So it means that by adding functionally important variants onto the high density SNP chip, you gain mapping precision because you need less amount of genome to explain this half of the heritability. In particular case, if you model your data in by using the coding variants or those finely mapped pleiotropic variants, you get further gain into the mapping precision by using base RC. So you can model you get a better results by modeling extra classes of variants for this particular two sets. So we further developed this genome-wide fine mapping approach related to Bayesian methods. And here I'm showing you the results of the mapping. So before the fine mapping, we have about 70 million sequence variants. So this is a Manhattan plot of GWAS showing that metric, the p-values of these 70 million SNPs. On the right-hand side, that's the GWAS results of the finely mapped 50,000 prioritized variants. You could see that if you just look at it by eyes, they don't look very different. That is because the peaks you're looking at mostly the same. And what really is different are these bottom ones, which are not very informative. So the graph on the right-hand side is much more thinned. So we used our fine mapping strategy to retain most of the good signals and remove the noise. And in fact, this is between the two graphs, we're talking about 200-fold marker reduction from 70 million to 50,000. So, and we then use this finely mapped set to customize a SNP chip that we could use in the dairy cattle genomic selection. So how is this finely mapped or customized SNP chip work in real time? So here we train our predictive model using our customized functional SNP chip using Australian cows, about 28,000, and we then predict them to predict traits in the New Zealand cow. So this is the actual cross-country genomic prediction. And here is the result of that prediction. So the y-axis is the genomic prediction accuracy correlation between the predicted phenotype or EBV and the real phenotype. This yellow bar is the accuracy of the customized functional SNP chip. The gray bar is the standard 50K chip. There is also a black bar, which is the prediction accuracy of the two panels combined together. You can see that across all the scenarios, the orange bar is always higher. That means it's better than the customized SNP chip, better predicts than the standard one. But also, when you combine the two together, it doesn't quite go over when you just use the customized chip alone. So it suggests that the customized functional SNP chip captured most of the important information and can deliver more accuracy. So we actually implemented in this functional SNP chip in the Australian dairy cattle breeding. So we have now already used this newly designed chip to genotype 15,000 Australian cattle up to that number. And we've tested the imputation of this panel and then how to refine it to match with what industry could use in their own genotyping and evaluation. because they have their own way. And here is a quick result showing that the increase of genomic prediction using the latest version of it basically shows that there is quite some increase of by using the new chip. But most importantly, we see increase of prediction accuracy in these crossbred animals, which are usually very difficult to predict. So that is really good results. So not just benefit the purebreds, but also the hybrids. Now, there's debate about EQT and QT, how do they overlap? In the previous study, we showed that if you look at the trait QTL from protein and fat percentage, and the overlap is about up to 5%. So that was really disappointing. But you can see that back then our EQTL sample size was about 120. Now, in our current ongoing work, we looked at this overlap again. So the QTL from protein and fat percentage, and by increase the sample size of EQTL mapping, here is about 3-4 folds, we have substantially increased the overlap between QTL and EQTL. So it means that the sample size or biological replicate or function data is really important. So that gives me a chance to conclude. So we show that functional information can improve genomic selection to assist breeding, and the benefits of using functional information increases with the growth of functional dataset. And we have more coming. There's FAANG, there's GTEx and roadmap, and so on. And we have issues doing that too. So we need more effective methods that we should develop to evaluate functional genomics data, to extract information from them, and so that we integrate that information into genomic selection. And also we need more industry-wise application of the functional genomics information, such as the development of the real data and products that the industry can benefit from this taxpayer-funded resource. So that is all I want to talk about. I'd like to thank all the people and funders that contributed by using the slide, and that's all. Thanks very much.
Thank you very much, Roy Dong. So we have again questions coming. It would be nice to tackle some of them because then there is a roundtable. So from Dialogue One is wondering what the conserved SNP mean in your context. If it means a SNPing cattle is still a SNPing human, for instance.
So the conserved sites are conserved. So basically a cutoff using a FAST-CON score across 100 species. So the 100 species, including from the fish to all the way to human.
And another question by Dominic, will the 50K functionally enriched variants work for beef cattle too?
Yeah, we haven't got the trait data set from beef cattle, but we are expecting to do that very soon. The functional data set should not matter too much, although it does a little bit in terms of what sort of breeds you use to get this functional data set.
And another question from Robert is, would such a functional panel perform for traits that do not have a major QTLs like feed efficiency?
Yeah, that is a really good question. So yeah. Now we mostly find that traits like milk production or other body size, they are easily find for EQT, but for traits like feed efficiency or something like fertility, that they are a bit difficult. So we're still working on that, but there are two issues around this matter. One is obviously the functional information. The second issue is the trait data set. Because of these traits, they're rather complex, they have low heritability, so we also need to increase the trait data set sample size to get a better estimate of the genetic architecture.
Great. Look, you accumulating questions because the talk was really nice. And I think we should close here and move to a round table. Thank you again.